Welcome to Cosmic Whispers, where cosmic mysteries come alive. Your journey beyond the stars begins now. In the autumn of 1928, Alexander Fleming returned to his cluttered laboratory at St. Mary's Hospital in London after a brief vacation. What he discovered would fundamentally alter the trajectory of human medicine and potentially hold the key to preventing future global pandemics. Among the petri dishes scattered across his workbench, one stood out with an unusual pattern that would soon capture the attention of the entire scientific world. Fleming had been studying Staphylococcus, bacteria, the same microorganisms responsible for dangerous infections that claimed countless lives in pre-antibiotic era. His laboratory practices were notoriously messy, a trait that colleagues often criticized. However, this apparent flaw would prove to be serendipitous. During his absence, a spore of Penicillium notatum mold had somehow contaminated one of his bacterial cultures. Rather than discarding the contaminated dish, Fleming's trained eye noticed something extraordinary. Around the greenish-blue mold, the bacteria had completely disappeared. The Staphylococcus colonies that should have formed a continuous lawn across the agar surface showed clear zones of inhibition surrounding the fungal growth. This observation represented more than mere contamination. It suggested that the mold was producing a substance capable of killing or inhibiting bacterial growth. Fleming's scientific curiosity was immediately piqued, recognizing that this phenomenon deserved serious investigation. The Scottish bacteriologist began systematic experiments to understand this mysterious antibacterial effect. He cultured the mold in pure form and discovered that its liquid medium possessed remarkable properties against various bacterial species. Fleming named this active substance penicillin after the Penicillium genus of the producing mold. His initial tests revealed that penicillin was effective against numerous gram-positive bacteria, including Streptococcus, Staphylococcus, and Pneumococcus species. Every great journey starts with a few brave explorers. Right now, Cosmic Whispers is just beginning, and your support means everything. If you enjoy unraveling the mysteries of the universe, hit subscribe. It's free and it helps us grow this community together. Fleming's discovery occurred during an era when bacterial infections represented humanity's greatest medical threat. Pneumonia, sepsis, and wound infections routinely killed patients who would be easily treated today. The mortality rate from bacterial pneumonia exceeded 30%, while post-surgical infections claimed lives with frightening regularity. Fleming recognized that his accidental discovery might hold the solution to these devastating medical challenges. However, Fleming faced significant obstacles in developing penicillin into a practical therapeutic agent. The mold produced relatively small quantities of the active compound, and purification techniques were primitive by modern standards. Additionally, the scientific community initially showed limited interest in his findings. Fleming published his results in the British Journal of Experimental Pathology in 1929, but the paper received minimal attention from his peers. Despite these challenges, Fleming continued his research, though progress remained slow. He demonstrated that penicillin was non-toxic to human cells and showed promise in treating localized infections. However, the technical difficulties in large-scale production and purification prevented widespread clinical application. Fleming's work remained largely theoretical until the early 1940s, when Howard Florey and Ernst Boris Chain recognized its potential and developed methods for mass production. The question that emerges from Fleming's revolutionary discovery extends far beyond historical curiosity. Could the principles underlying penicillin's development provide a roadmap for combating future pandemics? As we face an increasingly interconnected world where novel pathogens can spread globally within days, understanding how Fleming's accidental discovery led to one of medicine's greatest triumphs becomes crucial for pandemic preparedness strategies. This exploration will examine how Fleming's methodical approach to studying an unexpected observation created the foundation for modern antibiotic therapy, and whether similar scientific principles could be applied to develop rapid response systems for future pandemic threats. Understanding how penicillin functions at the molecular level reveals the elegant simplicity behind one of medicine's most powerful weapons against bacterial infection. The mechanism of action involves disrupting fundamental processes that bacteria require for survival and reproduction, specifically targeting the construction and maintenance of their protective cell walls. Bacterial cell walls consist primarily of peptidoglycan, a complex polymer that provides structural integrity and shape to bacterial cells. This peptidoglycan layer is significantly thicker in gram-positive bacteria, which explains why penicillin shows particular effectiveness against organisms like Staphylococcus and Streptococcus species. The peptidoglycan structure resembles a molecular mesh with long chains of sugar molecules cross-linked by short peptide bridges. The biosynthesis of peptidoglycan involves several enzymatic steps, with the final cross-linking process being particularly crucial. Transpeptidase enzymes, also known as penicillin-binding proteins, catalyze the formation of peptide cross-links that stabilize the cell wall structure. These enzymes recognize specific amino acid sequences in the peptidoglycan precursors and facilitate the chemical bonds that hold the entire structure together. Penicillin's molecular structure contains a beta-lactam ring, a four-membered cyclic amide that serves as the antibiotic's active component. This ring structure closely resembles the natural substrate that transpeptidase enzymes normally process during cell wall synthesis. When penicillin encounters these enzymes, the beta-lactam ring irreversibly binds to the active site, effectively blocking the enzyme's normal function. This irreversible inhibition prevents bacteria from completing cell wall synthesis, particularly during periods of active growth and division. As bacterial cells attempt to reproduce, they cannot form the cross-linked peptidoglycan structure necessary for maintaining cellular integrity. The result is weakened cell walls that cannot withstand the osmotic pressure differences between the bacterial interior and external environment. Without proper cell wall function, bacterial cells become vulnerable to osmotic lysis. Water enters the cell through osmosis faster than the compromised cell wall can compensate, causing the bacterial cell to swell and eventually burst. This bactericidal effect explains why penicillin is particularly effective against rapidly dividing bacteria, as these organisms are actively synthesizing new cell wall material. The selectivity of penicillin for bacterial cells over human cells stems from fundamental differences in cellular structure. Human cells lack peptidoglycan cell walls entirely, instead relying on flexible cell membranes for structural integrity. Since penicillin specifically targets peptidoglycan synthesis enzymes, it has minimal direct effects on human cellular processes, contributing to its relatively low toxicity profile. Fleming's original observations captured this selective toxicity perfectly. His initial experiments demonstrated that penicillin concentrations effective against bacteria showed no harmful effects on human white blood cells or tissues. This therapeutic window between antibacterial efficacy and human toxicity represents one of penicillin's most valuable characteristics for clinical application. The molecular specificity of penicillin's action also explains patterns of bacterial resistance that would later emerge. Bacteria can develop resistance through several mechanisms, including production of beta-lactamase enzymes that break down the beta-lactam ring before it can inhibit transpeptidases. Other resistance mechanisms involve modifications to penicillin, binding proteins that reduce the antibiotic's binding affinity. Understanding these molecular details provides crucial insights for developing strategies against future pandemic threats. The principle of targeting pathogen-specific processes while sparing host cells represents a fundamental approach that could be applied to antiviral drug development. Just as penicillin exploits differences between bacterial and human cellular machinery, effective pandemic countermeasures might target viral replication processes that have no human counterparts. Modern drug discovery programs increasingly rely on detailed molecular understanding to identify promising therapeutic targets. High-throughput screening techniques can rapidly test thousands of compounds against specific pathogen enzymes, potentially accelerating the timeline from discovery to clinical application. This systematic approach builds upon the foundation that Fleming established, through careful observation and methodical experimentation. The success of penicillin demonstrates how understanding fundamental biological processes can lead to transformative medical interventions, providing a model for approaching future pandemic preparedness challenges. The journey from Fleming's laboratory observation to widespread clinical application of penicillin required overcoming enormous technical and logistical challenges that nearly derailed the entire enterprise. While Fleming's initial discovery demonstrated penicillin's antibacterial properties, transforming this laboratory curiosity into a life-saving medicine demanded innovations in biochemistry, engineering, and industrial manufacturing that pushed the boundaries of 1940s technology. Howard Florey and Ernst Boris Chain at Oxford University recognized penicillin's therapeutic potential and began systematic efforts to develop practical production methods in 1939. Their team faced immediate obstacles in purifying and concentrating the active compound from crude mold cultures. Fleming's original methods yielded extremely dilute penicillin solutions that were insufficient for treating serious infections in humans. The Oxford researchers developed improved cultivation techniques using specialized media that enhanced mold growth and penicillin production. They discovered that Penicillium chrysogenum, a variant strain, produced significantly higher yields than Fleming's original Penicillium notatum. However, even with optimized conditions, laboratory-scale production remained woefully inadequate. For clinical trials, let alone widespread therapeutic use, early purification attempts relied on organic solvent extraction and primitive chromatographic techniques. The researchers used ether and chloroform to extract penicillin from aqueous mold cultures, followed by crystallization procedures to obtain partially purified material. These methods were labor-intensive, inefficient, and produced inconsistent results. Batch-to-batch variability complicated dosage determination and clinical evaluation. The first successful clinical trials occurred in 1941, when Florey's team treated several patients with severe infections using their limited penicillin supplies. The results were dramatic, with patients recovering from conditions that would have been fatal without treatment. However, the researchers could produce only enough penicillin to treat a handful of cases, highlighting the urgent need for scaled-up manufacturing capabilities. Recognizing that European production capacity was insufficient during wartime, Florey and his colleague Norman Heatley traveled to the United States in 1941 to seek American assistance in developing large-scale penicillin production. They initially approached the U.S. Department of Agriculture's Northern Regional Research Laboratory in Peoria, Illinois, where researchers had expertise in fermentation processes for industrial applications. American scientists made crucial contributions to solving the production challenge. They developed deep tank fermentation methods that replaced the surface culture techniques used in early production. These submerged fermentation systems allowed much larger-scale cultivation using standard industrial equipment. The addition of corn steep liquor, a byproduct of corn processing, dramatically increased penicillin yields by providing essential nutrients for mold growth. The development of penicillin production became a collaborative effort involving multiple pharmaceutical companies, government agencies, and academic institutions. Merck, Pfizer, Abbott, and other companies contributed expertise in fermentation technology, purification methods, and quality control procedures. This unprecedented cooperation between competing organizations demonstrated the potential for rapid innovation when addressing urgent public health needs. Industrial-scale production required solving numerous technical challenges beyond basic fermentation. Maintaining sterile conditions in large vessels, controlling temperature and pH throughout extended fermentation periods, and developing efficient downstream processing methods all demanded engineering innovations. Companies invested heavily in specialized equipment and facilities designed specifically for antibiotic production. Quality control became increasingly important as production volumes expanded. Standardized potency assays ensured consistent therapeutic effectiveness across different batches and manufacturers. The development of international units for measuring penicillin activity, allowed accurate dosing and facilitated global distribution of supplies. By 1943, American pharmaceutical companies were producing sufficient quantities of penicillin to supply military medical needs and begin civilian distribution. Production increased from millions of units annually in early years to billions of units by the war's end. This scaling-up process demonstrated how fundamental scientific discoveries could be rapidly translated into practical medical interventions when adequate resources and coordination were applied. The transformation of penicillin from laboratory observation to mass-produced medicine established principles for pharmaceutical development that remain relevant for pandemic preparedness. The combination of academic research, government support, and industrial expertise created a model for rapid response to emerging health threats that could inform strategies for addressing future pandemic challenges. The introduction of penicillin into clinical practice during the 1940s ushered in what medical historians now recognize as the golden age of antibiotics, fundamentally transforming medical practice and dramatically reducing mortality from bacterial infections. However, this revolutionary period also contained the seeds of a future crisis that would challenge the very foundations of antibiotic therapy and provide crucial lessons for pandemic preparedness strategies. Penicillin's clinical impact was immediately apparent and statistically overwhelming. Mortality rates from pneumococcal pneumonia dropped from approximately 30% to less than 5% within years of widespread penicillin availability. Puerperal sepsis, the leading cause of maternal death during childbirth, became largely preventable. Surgical procedures that had previously carried prohibitive infection risks became routine, enabling advances in cardiac surgery, organ transplantation, and other complex medical interventions. The societal implications extended far beyond individual patient outcomes. Hospitals could perform more aggressive treatments, knowing that post-operative infections were manageable. Military medicine was revolutionized, with battlefield wound infections no longer representing automatic death sentences for injured soldiers. Public health officials observed significant decreases in deaths from scarlet fever, rheumatic fever, and other streptococcal diseases that had previously claimed thousands of lives annually. However, Alexander Fleming himself had predicted the eventual emergence of antibiotic resistance in his Nobel Prize acceptance speech in 1945. Fleming warned that inadequate dosing or premature discontinuation of treatment could select for resistant bacterial strains, potentially rendering penicillin ineffective over time. His prescient observations were largely overlooked amid the euphoria surrounding penicillin's miraculous therapeutic effects. The first documented cases of penicillin resistance appeared within months of widespread clinical use. Hospital laboratories began isolating Staphylococcus aureus strains that showed reduced susceptibility to penicillin concentrations that had previously been uniformly effective. These resistant organisms were initially confined to hospital environments where antibiotic use was most intensive, but gradually spread to community settings. Bacterial resistance mechanisms proved remarkably diverse and sophisticated. Many bacteria developed beta-lactamase enzymes capable of breaking down penicillin's active beta-lactam ring before it could inhibit cell wall synthesis. Other organisms modified their penicillin-binding proteins, reducing the antibiotic's ability to interfere with peptidoglycan formation. Some bacteria developed efflux pumps that actively expelled penicillin from their cellular interior. The speed of resistance development surprised many researchers who had assumed that bacterial adaptation would require much longer timeframes. Within a decade of penicillin's introduction, resistant strains had become common in many hospital settings. By the 1960s, penicillin resistance among Staphylococcus aureus exceeded 50% in many locations, forcing clinicians to seek alternative therapeutic approaches. Pharmaceutical companies responded to emerging resistance by developing semi-synthetic penicillin derivatives designed to overcome specific resistance mechanisms. Methicillin, developed in 1959, remained effective against penicillin-resistant Staphylococcus strains by avoiding degradation by common beta-lactamase enzymes. However, methicillin-resistant Staphylococcus aureus strains emerged within years, demonstrating bacteria's remarkable capacity for adaptation. The broader antibiotic resistance phenomenon extended beyond penicillin to include virtually all antimicrobial agents as they were introduced. Streptomycin resistance appeared shortly after its clinical introduction, followed by resistance to chloramphenicol, tetracycline, and other antibiotics. Each new antibiotic seemed to trigger corresponding resistance mechanisms, creating an ongoing evolutionary arms race between pharmaceutical development and bacterial adaptation. Agricultural use of antibiotics as growth promoters and prophylactic agents accelerated resistance development by creating selective pressure across vast populations of commensal bacteria. Low-level antibiotic exposure in livestock selected for resistant strains that could subsequently transfer resistance genes to human pathogens through horizontal gene transfer mechanisms. The overuse and misuse of antibiotics in human medicine contributed significantly to resistance development. Patients who failed to complete prescribed courses allowed partially resistant bacteria to survive and multiply. Inappropriate prescribing for viral infections provided no therapeutic benefit while continuing to select for resistant bacterial strains in normal flora. Global travel and trade facilitated the worldwide spread of resistant organisms, transforming local resistance problems into international challenges. Resistant bacterial strains could cross continents within days, establishing themselves in new populations before public health authorities recognized their presence. The penicillin resistance story provides crucial insights for pandemic preparedness strategies. The rapid development and global spread of resistance demonstrates how pathogens can adapt to therapeutic interventions faster than anticipated. Understanding these evolutionary pressures becomes essential for developing sustainable approaches to pandemic control that account for pathogen adaptation over time. Modern pandemic planning must incorporate lessons learned from antibiotic resistance, including the importance of judicious use, combination therapies, and continuous surveillance for emerging resistance patterns. The antibiotic discovery landscape of the 21st century presents a stark contrast to the golden age of the 1940s and 50s, when researchers regularly identified new classes of antimicrobial compounds with relative ease. Today's pharmaceutical scientists face unprecedented challenges in developing novel antibiotics, creating a critical gap between medical need and therapeutic innovation that has profound implications for pandemic preparedness strategies. The deceleration in antibiotic discovery reflects several converging factors that have fundamentally altered the research environment. Natural product screening, which yielded penicillin and most other major antibiotic classes, has encountered what researchers describe as the low-hanging fruit problem. Decades of intensive screening have identified most easily discoverable antimicrobial compounds from soil microorganisms, marine environments, and other traditional sources. Modern high-throughput screening technologies can test millions of synthetic compounds against bacterial targets, yet this approach has yielded surprisingly few clinically viable antibiotics. The complexity of bacterial physiology means that compounds showing activity in laboratory assays often fail to demonstrate effectiveness in living organisms. Issues with drug penetration, metabolism, toxicity, and pharmacokinetics eliminate most promising candidates before they reach clinical testing. The economic incentives for antibiotic development have become increasingly unfavorable compared to other therapeutic areas. Antibiotics are typically used for short treatment courses, limiting revenue potential compared to chronic disease medications that patients take for years or decades. Additionally, new antibiotics are often reserved as last resort treatments to preserve their effectiveness, further reducing market opportunities for manufacturers. Regulatory requirements for antibiotic approval have become more stringent following safety concerns with several compounds that showed unexpected toxicities. During clinical development, the cost of bringing a new antibiotic through all phases of clinical testing now exceeds $1 billion, with success rates remaining disappointingly low. Many pharmaceutical companies have abandoned antibiotic research programs entirely, focusing resources on more profitable therapeutic areas. The scientific challenges in antibiotic development reflect the sophisticated defense mechanisms that bacteria have evolved over billions of years. Unlike the relatively simple targets that Fleming's penicillin exploited, modern drug developers must contend with multiple resistance mechanisms, efflux pumps, and bacterial biofilms that provide protection against antimicrobial agents. Target identification has become increasingly difficult as researchers exhaust obvious bacterial vulnerabilities. Essential bacterial processes that differ sufficiently from human cellular functions to allow selective targeting are relatively rare. Many promising targets identified through genomic studies have proven unsuitable for drug development due to technical challenges in finding compounds that effectively interact with these proteins. The rise of multi-drug-resistant organisms has complicated clinical trial design and regulatory evaluation. Traditional antibiotic trials compare new drugs to existing standard treatments, but this approach becomes problematic when standard treatments are largely ineffective against resistant pathogens. Conducting placebo-controlled trials raises ethical concerns when patients have life-threatening infections. Global regulatory harmonization remains incomplete, requiring companies to navigate different approval processes across major markets. Variations in clinical trial requirements, efficacy endpoints, and safety standards increase development costs and timelines. These regulatory complexities particularly affect smaller companies and academic institutions that lack resources for international development programs. The decline in academic antibiotic research has reduced the pipeline of early-stage discoveries that previously fed pharmaceutical development programs. Many universities have reduced or eliminated medicinal chemistry and microbiology programs, while funding agencies have shifted priorities toward other research areas. The loss of expertise in antibiotic development has created knowledge gaps that further impede progress. Alternative approaches to traditional antibiotic development are emerging from these challenges. Bacteriophage therapy, which uses viruses that specifically target bacterial pathogens, offers potential advantages, including host specificity and reduced resistance development. Immunomodulatory approaches that enhance host defenses rather than directly killing pathogens represent another promising direction. Antimicrobial peptides produced by various organisms, including humans, provide templates for developing new therapeutic agents. These naturally occurring compounds often have novel mechanisms of action that differ from conventional antibiotics, potentially overcoming existing resistance mechanisms. However, peptide drugs face unique challenges in terms of stability, delivery, and manufacturing costs. Combination therapy strategies that use existing antibiotics in novel ways can potentially restore effectiveness against resistant organisms. Adjuvant compounds that disable bacterial resistance mechanisms when used alongside conventional antibiotics offer another approach to extending the useful life of existing drugs. The parallels between antibiotic development challenges and pandemic preparedness become apparent when considering the need for rapid response capabilities. The current antibiotic discovery timeline, often requiring 15 to 20 years from initial discovery to clinical approval, would be completely inadequate for addressing pandemic threats. Understanding why antibiotic development has slowed provides insights into potential obstacles for developing pandemic countermeasures and suggests strategies for overcoming these barriers through alternative approaches and policy interventions. The principles underlying Fleming's penicillin discovery and its subsequent development offer profound insights for modern pandemic preparedness strategies, particularly regarding the identification, development, and deployment of medical countermeasures against emerging infectious disease threats. Analyzing the penicillin story through the lens of contemporary pandemic challenges reveals both opportunities and obstacles that must be addressed to improve global response capabilities. Fleming's initial discovery exemplifies the importance of maintaining robust fundamental research programs that can respond rapidly when unexpected observations emerge. His ability to recognize the significance of bacterial inhibition around contaminating mold reflected years of training in microbiology and systematic experimental approaches. Modern pandemic preparedness requires similar investments in basic virology, immunology, and infectious disease research that provide the knowledge foundation for rapid response when novel pathogens emerge. The observational skills that allowed Fleming to notice unusual patterns in his contaminated cultures translate directly to modern disease surveillance systems. Just as Fleming's trained eye detected bacterial inhibition zones, contemporary epidemiologists must maintain vigilant monitoring for unusual disease patterns that might signal emerging pandemic threats. Investment in global surveillance networks, diagnostic capabilities, and data-sharing systems represents the modern equivalent of Fleming's careful laboratory observations. The systematic approach that Fleming used to characterize penicillin's properties provides a template for rapid pathogen characterization during pandemic responses. Fleming methodically tested penicillin against various bacterial species, determined optimal growth conditions for the producing mold, and evaluated toxicity in laboratory studies. Modern pandemic response protocols should similarly emphasize rapid but thorough characterization of novel pathogens, including their transmission mechanisms, pathogenicity factors, and potential therapeutic targets. The collaboration between Fleming's initial discovery and the Oxford team's development efforts demonstrates the critical importance of multidisciplinary cooperation in translating scientific observations into practical medical interventions. Florey and Chain brought expertise in biochemistry and pharmacology that Fleming lacked, enabling the purification and clinical evaluation that Fleming could not accomplish alone. Pandemic preparedness strategies must similarly foster collaboration between academic researchers, pharmaceutical companies, and government agencies to accelerate countermeasure development. The transition from laboratory scale to industrial production of penicillin illustrates the manufacturing challenges that pandemic response must address. The technical innovations required for large-scale fermentation, purification, and quality control took several years to develop, even with unprecedented cooperation and resources. Modern pandemic preparedness must include pre-positioned manufacturing, capabilities, flexible production platforms, and supply chain redundancies that can be rapidly scaled when needed. The international cooperation that enabled penicillin's wartime production provides a model for global pandemic response coordination. The collaboration between British researchers and American pharmaceutical companies demonstrated how sharing scientific knowledge and manufacturing expertise can accelerate development timelines. Contemporary pandemic preparedness requires similar mechanisms for international cooperation, including intellectual property frameworks that facilitate rapid knowledge sharing while maintaining innovation incentives. The emergence of penicillin resistance offers cautionary lessons about pathogen adaptation that directly apply to pandemic countermeasure development. Fleming's early warnings about resistance development were largely ignored until resistant strains became widespread, demonstrating the importance of incorporating evolutionary considerations into pandemic response strategies. Modern pandemic planning must account for pathogen adaptation and include strategies for maintaining countermeasure effectiveness over time. The economic challenges that have slowed modern antibiotic development mirror potential obstacles to pandemic countermeasure development. Just as pharmaceutical companies have reduced antibiotic research due to limited profitability, pandemic preparedness investments may suffer from similar market failures. Public-private partnerships, advance purchase commitments, and other policy mechanisms can address these economic barriers by ensuring adequate returns on pandemic preparedness investments. The regulatory frameworks developed for antibiotic approval provide precedents for expedited review processes during public health emergencies. The wartime acceleration of penicillin development and approval demonstrated that regulatory flexibility can significantly reduce development timelines without compromising safety. Modern emergency use authorization procedures build on these precedents while maintaining appropriate safety standards. The global distribution challenges encountered during early penicillin deployment highlight equity considerations that remain central to pandemic preparedness. Initial penicillin supplies were primarily allocated to military uses and wealthy patients, leaving vulnerable populations without access to life-saving treatment. Contemporary pandemic preparedness must incorporate equity considerations from the planning stage to ensure that countermeasures reach all affected populations regardless of economic status or geographic location. Platform technologies that can be rapidly adapted for different pathogens represent a modern evolution of the flexible production capabilities developed for penicillin. Messenger RNA vaccine platforms, for example, can potentially be reconfigured for new pathogens within months rather than years, providing the rapid response capability that pandemic threats require. The integration of these lessons from penicillin's development into modern pandemic preparedness strategies offers the potential to significantly improve global response capabilities while avoiding the pitfalls that have limited antibiotic development in recent decades. The convergence of artificial intelligence, advanced biotechnology, and precision medicine approaches is creating unprecedented opportunities for developing medical countermeasures that could revolutionize pandemic preparedness and response capabilities. These emerging technologies build upon the fundamental principles established by Fleming's penicillin discovery, while addressing the complex challenges that characterize 21st-century infectious disease threats. Artificial intelligence-driven drug discovery platforms are beginning to accelerate the identification of novel therapeutic compounds in ways that could dramatically compress development timelines. Machine learning algorithms can analyze vast databases of molecular structures, predict drug target interactions, and identify promising compounds from millions of possibilities within hours rather than years. These AI systems learn from successful drug development patterns and can suggest modifications to existing compounds that might enhance their effectiveness against specific pathogens. DeepMind's AlphaFold protein structure prediction system exemplifies how AI can provide insights that would be impossible through traditional experimental approaches. By accurately predicting three-dimensional protein structures from amino acid sequences, AlphaFold enables researchers to identify potential drug binding sites and design targeted therapeutics without lengthy crystallography studies. This capability could prove invaluable for rapidly developing countermeasures against novel pandemic pathogens whose structures have never been experimentally determined. Computational modeling of pathogen evolution represents another frontier where AI technologies can inform countermeasure development strategies. Machine learning systems can analyze pathogen genomic sequences to predict likely evolutionary pathways and anticipate resistance development before it occurs clinically. This predictive capability could enable the development of combination therapies and next-generation countermeasures that remain effective even as pathogens adapt. Platform technologies for rapid countermeasure development are reaching maturity in several therapeutic areas. Messenger RNA vaccine platforms demonstrated their potential during the COVID-19 pandemic, enabling vaccine development and deployment within months rather than the typical decade-long timeline. Similar platform approaches are being developed for monoclonal antibody production, antiviral drug synthesis, and diagnostic test development. Synthetic biology approaches offer the potential to engineer biological systems specifically designed for pandemic countermeasure production. Engineered microorganisms could be programmed to produce specific therapeutic compounds rapidly and efficiently, potentially enabling distributed manufacturing that reduces supply chain vulnerabilities. These synthetic biology platforms could be particularly valuable for producing complex biological therapeutics that are difficult to synthesize through traditional chemical methods. Personalized medicine approaches are beginning to influence pandemic preparedness strategies through pharmacogenomic considerations and individualized risk assessment. Understanding how genetic variations affect drug metabolism and immune responses can inform dosing strategies and identify individuals who might require alternative treatments. This precision medicine approach could improve countermeasure effectiveness while reducing adverse effects. Nanotechnology applications in drug delivery are creating new possibilities for targeted therapeutic delivery and enhanced bioavailability. Nanoparticle formulations can improve drug stability, enable controlled release, and facilitate delivery to specific tissues or cellular compartments. These advanced delivery systems could prove particularly valuable for antiviral drugs that must reach specific anatomical sites to be effective. Point-of-care diagnostic technologies that provide rapid, accurate pathogen identification are becoming increasingly sophisticated and accessible. These portable diagnostic systems could enable immediate implementation of targeted countermeasures rather than empirical treatment approaches. Integration of diagnostic and therapeutic capabilities into single platforms could further streamline pandemic response procedures. Global manufacturing networks with distributed production capabilities represent a strategic approach to ensuring countermeasure availability during pandemic situations. Rather than relying on centralized production facilities that can become bottlenecks during crises, distributed manufacturing networks could provide redundancy and reduce supply chain vulnerabilities. Advanced manufacturing technologies, including continuous production processes and automated quality control systems, could enable rapid scaling of production capacity. Regulatory science innovations are creating pathways for accelerated countermeasure approval while maintaining safety standards. Adaptive clinical trial designs, real-world evidence collection, and model-informed drug development approaches can reduce development timelines while generating robust safety and efficacy data. These regulatory innovations build upon lessons learned from emergency use authorization procedures during recent public health emergencies. International cooperation frameworks for pandemic countermeasure development are evolving to address intellectual property, technology transfer, and equitable access challenges that have limited response effectiveness in the past. Advanced purchase commitments, patent pooling arrangements, and technology sharing agreements can align commercial incentives with public health needs while ensuring innovation continues. The integration of these emerging technologies with lessons learned from Fleming's penicillin discovery creates the potential for transformative improvements in pandemic preparedness capabilities. However, realizing this potential requires sustained investment in research and development, international cooperation, and regulatory frameworks that can accommodate innovative approaches while maintaining appropriate safety standards. The next breakthrough comparable to Fleming's penicillin discovery may not come from a single accidental observation, but rather from the systematic application of advanced technologies to pandemic preparedness challenges. By combining the observational skills and systematic approach that characterized Fleming's work with 21st century technological capabilities, the scientific community may be positioned to develop countermeasures that can prevent future pandemics from reaching the devastating scale of past infectious disease outbreaks. The ultimate goal remains the same as Fleming's original vision: transforming chance observations into life-saving medical interventions that protect human health. On a global scale,